Good morning. Good day, everyone. We're all welcome. It's past 4 p.m. Nigerian time, so good evening to everyone. And we are all welcome to this live session. As a way of signing in, please, you can drop your name and where you're joining us from, so we can just do a quick introduction of ourselves. We're all welcome once again. So let's just do that in the next 2 to 3 minutes as we get started um, in this session this evening. So I will be checking through the comments, the welcomers. Okay, I see Botede, you're welcome. I see Adaobi from Nigeria, you're welcome. Thank you for joining us this evening. I see Adeboji Bodwin from Nigeria, thank you so much. Festus from Nigeria, Colin from Ghana, it's good to have you on board. I see Ulakem from Nigeria, thank you for joining. I see Chimuka, I'm sorry if I didn't get that right, from Zambia, you're welcome. I see you. Shepard from Nigeria, you're welcome. Thank you so much. Okay, I'm not from Nigeria. Thank you for joining us. Blessing Ayane from Nigeria. Good to have you on board. Philip from Nigeria. Thank you so much. Dr. Oyelakin from Ilorin, Nigeria. Thank you for joining us this wonderful evening. Let's keep it coming. Let's keep it coming. And while we wait for more people to join us this evening, Adela from Nigeria, thank you. Sophia from Nigeria, Agamu from Abuja. It's good to have you. Thank you for joining us. Thank you for joining us. Thank you to everyone for being on this call right here at this moment. We are so excited to have you, and we know that you are going to have an exciting and amazing time with us this evening. Justina from Nigeria, welcome. Alice from Nigeria, I see. Thank you so much for joining. So, for everyone that is on this call right now, I would quickly want us to do something for me right now, and it's quite simple. It's just to have more people join us before we kickstart the session fully this evening. So, what I want you to do for me is to just copy the live link of this session and go share it with your community, your friends, share it with whoever is meant to be here that you know is supposed to be here. Just share it, and once you're done, come and type done in the chat box so that I can know that we have some people that have done that already. So let's just quickly do that um, in the next few minutes. Let's share the link with our friends, our community. Let's get more people on board. Let's have more people in. Okay, I see more people signing in. Anna from Nigeria, thank you for joining. Omar from Nigeria, Victor Paul, you're welcome. Good to have you on board. Mohammed, you are most welcome. So please, let's quickly do what I asked earlier. Let's share this live link with our friends and our community, and let's get more people right here on this call. Let's get as much people as we can right here on this call. Be rest assured that you are going to have, a wonderful time this evening. You're going to learn so much, and you are going to be. It's going to be a worthwhile time for every one of us. So that's why we want to get more people right here on the call. So please let's join us to make impact. Let's reach more people with this. let's reach more people with this live stream session, and let's have more people on board. Thank you so much as you do that. I'm still expecting. Okay, I see Godwin is done. Thank you so much for sharing the link. I expect more people to do that. Please let's have more people share the link. At least I need 5 people's responses to tell me that they've shared the link before we continue the session this evening. So please, if you've not shared the live link, make sure you go right ahead and do that as soon as possible. Okay, thank you so much, everyone. I see our responses coming in. Thank you so much, everyone. Okay, thank you for sharing the link. Thank you so much. So this evening we already have our speaker on board, and she's quite ready to teach us, to enlighten us, and to bring us up to speed on transcriptomics. As you might have seen on the flyer, today we are going to be talking on the tools and techniques required or needed for transcriptome profiling. So, for everyone who has been looking into delving into the field of transcriptomics, or you just been hearing the word, you really don't know what it's all about. So be rest assured that this evening you are going to be getting a lot of your questions answered as our speaker comes right on board. So before we begin fully, a few housekeeping rules for everyone. So just wanted to tell us that you might need to get your writing materials, maybe your phone or maybe a note and a pen, just to make sure that you are keeping track of your. Yes, so you might want to get your writing material. You want to stay undistracted. We are probably going to be here for next 60 to 90 minutes, however it will take. So you want to stay undistracted, and you want to have your writing material close to you. And also you might want to note your questions down as the discussion might be going on. There will be a question and answer phase after the speaker is done with our presentation. And finally, as we have already announced or said earlier, that only people that join this live call will be getting certificates. So in lieu of that, before the end of the call, a link would be sent in the chat box for you to fill, and then your certificates will be sent to your respective emails. So you need to ensure that you stay till the end of the session if you really want to get a certificate. So that's how you'll be getting your certificate this time around. So please ensure that you stay till the very end of the meeting, because that is when you'll be able to get the link for you to fill to be able to get your certificate. So I hope that is well understood. Okay, so I'll be bringing up our speaker on board right about now. Like I would always say that at Genomark Institute Incorporated, we are committed to your growth, and we are quite committed to making impact in the lives of bioinformaticians, anyone in life sciences field, and all of that. So there's always something for you here, right? Whether you're just starting out your journey, or you've gotten a hang of it somehow, or you are even a professional already, there's always something for you here at Genomax, and that is why we always encourage you to stay connected with us. So, and that's why we are having, this session today, because we know that it's going to help, each and every one of us in our respective journey. So this evening, we're bringing up our speaker now, and by name, she is Mutumaya Ilisomi, and should be the one facilitating this session today with us. She'll be taking us through the tools and techniques required for transcriptome profiling. So I would want you to give rapt attention as she, goes through the session. And like I said earlier, if you have questions, you can note them down, and then there will be a time for her to address those questions. So please, I want you to welcome with me this wonderful evening our speaker as she takes over right now. You're welcome, ma'am. It's good to have you.
Um, Good evening, everyone. Am I audible?
Yes, ma'am, you have.
And so, thank you very much for the opportunity to speak. I really appreciate it. So, I'm going to share my screen now to share my slide. Okay, as a form of introduction, my name is Mutunwa Elisomi. As it has been rightly said, I work with Genomax Services and Consorts. I'm the team lead at Genomax Services and Consorts, where we offer bioinformatics services to institutions, to researchers alike. Please, if you can see my screen, please let me know. Can you see my screen?
Yes, ma'am, I can see your screen. We can see your
Okay,
screen.
and it's in presentation mode, right?
Yes, yes.
Okay, thank you very much. Okay, so I will go right into our topic of discussion for this evening. The topic is data-driven biology, tools and techniques for transcriptome profiling. So our topic is actually one that is multi-layered. We have two keywords, which are data-driven biology and transcriptome profiling. So I will first talk about data-driven biology. So modern biology reached a turning point in 2003 with the completion of the Human Genome Project. I don't know if you are familiar with bioinformatics, you must have heard of the project before to some extent. So it was a project that was completed in 2003. It actually took them, I think, over a decade to be able to sequence the whole human genome, which means from they were able to sequencing means they were able to get the base sequence, so the ACG and T sequence from the beginning to the end of the human genome. If you're familiar with molecular biology, you know that genome is the molecular content of the cell. In the nucleus of any organism, we have the genome, which is also known as the nucleic acid or the DNA. So it contains nucleotide sequences. So we have A, G, C, and T for the representation of the four types of nucleotides, nucleic acids that nucleotides you can have in the nucleic acid. So that project was completed in 2003, and they were able to get the sequence of genome, the sequence of base human being, and that came with a consequence, and the consequence was big data. So they were able to generate over 100 to 200 gigabytes of data from that process. So you can also imagine it. Well, you cannot write that kind of thing because the human genome is at least 30 billion base pairs. I think 3 billion base pairs, the human genome. So you cannot write that by hand. You need to store it. And when they decided to store it, they realized that they had over 100 to 200 gigabytes of data. So it was a milestone in modern biology. So it gave rise to two major new fields, which are genomics and bioinformatics. So genomics talk about sequencing the genome, So if we could do it for one person, that means we could do it for several other people. And we knowing the content of the DNA is actually very, very germane as biomedical researchers, because every symptoms, every condition actually generates from the molecular biology. It generates from the reactions that happen at the molecular level, our replication, translation, transcription, and translation. Those three processes are actually what are like the back end. If you are, I like to represent it with, you know, this coding stuff. So they will say there is the back end and there is the front end. So those conditions that we see, back end, what is happening is the molecular reactions, which are replication, transcription, and translation, which are being dysregulated. That's what leads to different conditions that we see. So being able to sequence the genome actually was a major breakthrough, because now we can study what's happening at the back end. And if we understand what's happening at the back end, we can know how to attack what we have before us, which are the symptoms. So another field that arose as a result of that was bioinformatics. So if you are studying genomics, we have already identified that we'll be having big data that normal statistics cannot solve for us. You know, all these SPSS statistics, they cannot solve it for us because they are very, very large. So that then the field of bioinformatics also came in. So bioinformatics now analyzing the genomic data that we got from genomic studies using computational and statistical methods. So those are the two major pillars of bioinformatics: computation and statistics. So those two fields, genomics and bioinformatics together, they now make up what we call today data-driven biology. So modern biology today majorly is data-driven biology, and they arise as a result of those two fields, genomics and bioinformatics. So our next agenda, the next keyword in our topic is transcriptome profiling. So what's transcriptome profiling? So you must have, I believe you must have heard this before. That's why you joined this session. Okay, I've been hearing this transcriptomics, this transcriptome. What does it actually mean? So transcriptome profiling, transcriptome actually comes from the word transcription. So you know, when I was mentioning molecular biology, I mentioned three different reactions. I mentioned translation, replication, transcription, and translation. So that middle reaction, transcription, is what gives rise to transcriptome. So what is transcriptome profiling? Transcriptome profiling is essentially an extension of genomics and bioinformatics. So it is we going from genomics now. So genomics tells us the genes. You know, when you study the genome, what are you studying? You're studying genes. If from basic biology, we know that a gene is a segment of the genome that codes for a particular protein. So if you're studying the genome, that means we are studying several genes. We are identifying, okay, this gene is from codon 1 to stop. This gene is from base pair 1 to base pair 2000. So that's what genomics is all about. So we knowing that a gene is there is not enough. We need to know because I like to say this, your body is very, very conservative. So because you can, let's say for example, we all know we can dance, right? We can dance, but we are in this session now, we will not start dancing. Why? Because it is not needed at the moment. What is needed at the moment is for you to sit down and listen to what is being taught and take notes and maybe ask your question later. That is what is necessary in this particular time. So your body understands that. So you will not suddenly start dancing, even though you have the potential to dance. So that's exactly what happens to when we're talking about replication, transcription, and translation. So the gene are in the genome, but the body will not express all the genes at once. So we said already that a gene codes for a protein, which means when a gene is translated, what we get is a protein that goes ahead to carry out several other physiological processes in the body. But because we can produce this protein does not mean we have to produce it every time. So the body knows how to regulate gene expression such that what we now have from the genome, we now have the transcriptome. So the transcriptome is like a spreadsheet containing all the genes that are expressed per time. So this process as I said, is regulation. So it differs per time. It differs based on the time frame that you are currently as a human being. Maybe you are young, maybe you are old. It differs based on your environment. You know, when maybe you are abroad, maybe you are in Nigeria, there are some things that you will need in those environments that you don't need here. For example, you will need in abroad, it's cold there, so maybe you will need more of white fat in your skin. But Nigeria here is very, very hot, so it's absurd for your body to be creating that. So it will not express those genes. So that is what transcriptomics is all about. So we are studying, okay, we know that these genes exist in this person, but which one is this person expressing at this particular time that we are talking? So that's what transcriptomics does. So transcriptomics uses two major techniques to do this. There is sequencing and there is computation. So just like we have said, genomics and bioinformatics. So we sequence the genome, and we sequence the transcriptome, and we generate the data, and then the data goes on for analyzing. So applications of transcriptome profiling, we have biomarker discovery. So this is with the use of the use of transcriptome profiling. So let's say a person has a disease condition. So we need to know, okay, for example, we have two people. Somebody is constantly stooling, or somebody is totally normal. So we need to understand, okay, why is this person going to the toilet 10 times in 1 hour? What's wrong with this person? So that's the constant stooling is a symptom. So it's the front end. You need to understand what's going on at the back end. So that's when you now go on to, okay, let's analyze. Maybe you have done. That's when you say, okay, let's check at the molecular level. Maybe there is something wrong molecularly. So we already established that this person, normal human beings can stool, right? It's normal for a human being to go to the toilet. So let's say we all have the potential to go to the toilet. But if this person is going to the toilet haphazardly, we need to understand why. So it means that something is being done, but it's being done in an absorbed manner. It's being done in an uncontrolled manner. And most times it's always a gene that is being expressed at an uncontrolled manner. So if we can do a transcriptome profiling of that person, and we identify the gene that is being, and we identify genes that have been expressed as an uncontrolled manner, and we're able to link them through literature to going to the toilet, then we can say, okay, maybe this gene can be identified as a biomarker. So what's a biomarker? A biomarker is something that like a signal. So if you see it, you know that this thing is here. So all these disease conditions that we know, diabetics, hepatitis, hypertension, Alzheimer's, all of them have their biomarkers. So they have like a signal, so that if you do a transcriptome profiling of somebody and those genes are dysregulated, you can say for sure that this person has this condition. So transcriptome profiling helps us to identify biomarkers. It helps us to study drug responses. Somebody takes a drug, another person takes a drug, and then we are seeing that this person is already fine after 2 days, but this person is in 1 week and it's still the same thing. So what's going on? Why is this person reacting to this drug separately? We can know that through transcriptome profiling. We have disease mechanisms. This is very, very interesting. When you do transcriptome profiling, you can understand what's happening at the back end of every symptoms. Why is this, like, maybe it's because I studied biochemistry. So I tend to relate everything to pathways. So if something is happening, that means one pathway is being dysregulated. So we need to know. So disease mechanisms means we are trying to know which pathway is being affected, which pathway is not running. Because if you are able to profile your body and you know what your body is doing part time, you will be wowed. We always say, I'm very, very busy. I'm very, very busy. I'm doing this, I'm doing. But you know that your body is actually more busier than you. Very, very much busier than you. Reactions that are happening at milliseconds, at seconds, they are happening very, very fast, and you don't even realize this. It does not shout that, okay, I'm about to do this one, though. Okay, now is replication that is next. It does not tell you all of those things, but they are happening. So when something is happening, when we are seeing the symptoms, then that means the body is being the one pathway or the other is being compromised, and transcriptome profiling helps us to see all those things. So to the next slide, we have different approach for transcriptomics. We have bulk. So essentially, you must have been hearing of RNA-seq. If you have been familiar with transcriptomics, you must have been seeing RNA-seq analysis. So that's how we do transcriptome profiling. You know, if you want to do, let's say you must have heard of whole genome analysis too. That means you are sequencing the whole genome of a person, like I said earlier, like you are knowing all the genes that this person have. But when we talk about RNA, when we talk about transcriptomics, you don't need all of the genes that the person has. You only need the genes that are being expressed per time. So and if you understand from the biology of molecular biology, you know that from DNA will go to RNA, and from RNA will go to protein. So a transcription from DNA is what gives us RNA. So RNA does not contain all the genes. It contains the genes that are being expressed per time. So when you want to do transcriptome profiling, you don't need the DNA, you need the person's RNA. Yes, you need the person's RNA. So for sequencing, so that's where we got the word RNA-seq from, means RNA sequencing. So there are different types of RNA sequencing. We have bulk RNA sequencing, we have single cell RNA sequencing, and we have spatial transcriptomics. Currently, the most widely known method is bulk RNA sequencing. So this bulk, like the name suggests, it is bulk. Please, am I audible?
Yes, ma'am, you can go ahead. Yeah. Okay.
Hello, am I audible?
Yes, ma'am, you can go ahead. I can hear you now.
Since when have I been off, please? I hope I didn't take too much time offline.
No, not at all. You can just continue from where you stopped.
Okay, thank you very much. I'm so sorry. So, as I was saying, so we have different approaches for RNA sequencing. We have bulk RNA, we have single cell RNA-seq. So, bulk RNA, like the name suggests, means you are sequencing in bulk. So, in this kind of sequencing, you use the tissue that you are interested in. Let's say you are interested in the liver. You want to do RNA sequencing on the liver. Let me use something familiar. Let's say pancreas. Let's say somebody has diabetes and you want to sequence the RNA in the pancreas of the person to do a profiling, to do a transcriptome profiling of the person. In this case, you need to get the you need to get the RNA molecule from the person's pancreas. Whole pancreas. That's what bulk RNA-seq is, and it is very, very cost effective. It has well established pipelines. People know it's an household technique. Then we have the single cell RNA-seq. This is new, but it is not so new anymore. So it profiles gene expression at individual cell resolution. No, so now this is like mind blowing. It does not do bulk like tissue. It tells you what is going on in each cell. Do you know that even a cell, you cannot see it with your eyes? So you trying to get insight into what is happening in a single cell is, I don't know, but it is very, very mind-blowing for me. So it profiles a gene at single cell resolution. So in a cell, in an organ, you can have different cell generations or different cell types. You have, for example, I think in the pancreas, you have alpha cells, you have beta cells, I think, and stuff like that. So in the brain now, for example, you have microglia, you have neurons, you have glial cells, and all of those stuff. So when you are doing single cell RNA-seq, you are not getting transcriptomics of the brain alone. You are getting transcriptomics of the neuron, you are getting for the microglia, you are getting for the ganglia, and every other cell type that is in that organ. So that's very much more compared to bulk. So you can make much more assumption with that kind, because now we are not even talking in terms of you are not just haphazardly saying something. You are very much specific with your results. So although it is very nice, it is more expensive, and the computational and also computationally intensive, which means that how do I put it? Which means computational power, if you want to do single cell RNA-seq, is not the normal one. It is not the normal one. You can still use a PC, but it's not going to be a 8 gig PC or 4 gig PC. You just crash your PC. So they are very much advanced computational power for it. And then the final one is spatial transcriptomics. This one is still very much new. But what it does is that it measures gene expression while preserving tissue architecture. So now this one even tells you the location of each gene, each cell in the organ. So you can tell you, so you can know that okay, this transcriptomics is for the upper tissue or something. It's for the lower tissue or something. So it, like for example, now the brain, now we have different regions, the two hemispheres. So if you want to do spatial transcriptomics, that you are getting transcriptomics, you can decide to get transcriptomics only for the okay, so you can get for a particular region of the tissue and not everywhere. So that's what spatial transcriptomics is all about. So after now that we have understood what data-driven biology is, we understand how transcriptomics came into board, we understand what we can do with transcriptomics, we understand the different approaches that are applicable to transcriptomics. Now, I'd like us to go into the transcriptomics workflow. The transcriptomics workflow. So every analysis in bioinformatics has a workflow. Yes. So you cannot just come and say this is what I want to do and this is what I will do. No. There is always like a guideline that you follow. And that's why also actually what makes bioinformatics also kind of relatively simple, because you know what you want to do. Every time you want to do a transcriptomics you can do it over and over again, and nobody will question you. You just change a few things. You can say, okay, I don't want to do this kind of plot. I want to do another plot. You can do a lot of things. So once you know the workflow. So for this workflow, I'm going to be using a case study so that it will be more understandable rather. So our case study is a transcriptome profiling of the retina of AD patients using 5x FAD mouse models. So what's okay? AD simply means Alzheimer's disease. Alzheimer's disease is a type of dementia that happens to old people. Though we now have cases of early onset, which means people that are even young, maybe less than 50, 60 also have the condition. But majorly, it is for old people. Aging is a risk factor for it. So once somebody is aging, there is a high possibility that the person will develop Alzheimer's disease. So that's the case condition that we are using today. So the 5x FAD mouse model is a transgenic mouse model, which means a transgenic mouse is a mouse whose genome has been compromised to start expressing symptoms that are only found in human beings. For example, now normally mouse do not develop Alzheimer's disease, but there is a way you can take a mouse and tweak its genome so that it starts giving the symptoms of somebody that has Alzheimer's disease. So why do researchers do this? They do this because translational research is expensive, and it is actually quite what's it called, it's actually not ethical for you to use real human beings to be trying out stuffs. So you think of an idea and then you start grabbing people from the hospital and start injecting them with your stuff. So it is very, very not ethical. So what do you do first? First, you try out your disease, you try out your what's it called? Your maybe your therapeutic or your idea, anything you and for example, now you want to study disease condition. You might not have access to real human beings that actually have the disease condition. For example, now in Nigeria, the people that actually have Alzheimer's disease in Nigeria do not even know that they have Alzheimer's disease in Nigeria. Yeah, so you cannot go to an hospital and say, okay, I'm looking for Alzheimer's patient, so I want to do this disease condition. Oh, yeah, take questionnaire and fill. Oh. No, because because of the nature of what's it called? The nature of the health care system we have in Nigeria, with those things are not really what's it called? Not really available to a lot of people. So it's like the level of awareness is very, very low. So that's the thing. So in that case, and you still want to study this condition because it's actually real. So what you can do is that you can use a mouse model. So what you do is that you tweak the genome of the mouse model. So but already in literature, we already know the genes that is compromised in a person that has Alzheimer's disease. So you can tweak that gene in that person and that person and that in that mouse rather, and that mouse will start this developing the symptoms. So if you have a therapeutic you wanted to apply, you can start applying it to that person, to that mouse, and then you keep monitoring the condition. And you know that we human beings we are very, very close to all these animals too in terms of the genome, the genomic content of the person. So actually what we are saying is that the discoveries made in that kind of study can be not so completely generalized, but at least like 70%, 60% generalization can be applied to human beings. So in this what's it called? In this case study, our tissue is the retina. So we want to study for Alzheimer's patients. We discovered that Alzheimer's patients have what we call retinal pathology, which means they begin to have visual impairments as part of as part of the symptoms of their condition. They have visual impairments. Okay. So they have visual impairment as part of their disease condition. So what can we do? We want to study how the Alzheimer's disease affects the retina of a person. So what we do now is we design the first step in any workflow is that you design your experiments. You don't just go and start collecting data. You need to design your experiments. So the first thing you do, you define your hypothesis. For this kind of research now, our hypothesis is that Alzheimer's disease affects the retina of patients. So like Alzheimer's disease patients have retinal pathology. That's our hypothesis. So what do? We want to investigate this hypothesis. So we define our groups. We have the Y type, which is the control, and we have the disease condition, which is the 5XFAD model. So we define our group. You can say, okay, so this is when you begin to choose your biological replicates. So biological replicate simply means replicates that have the same condition. So for example, Y type. Why, for example, you have two groups in this experiment. In some experiments, you can have up to 3 groups, up to 4 groups in one experiment. But this experiment, we have 2 groups. So for each group, you cannot just use one mouse for each group and say, okay, I have one Y type animal and I have one FAD model and I want to start doing research. No, it's not possible. Even in normal research, you are advised to have at least 5 replicates or 4 replicates per group. Not now talk of a genomic experiment. A genomic experiment in terms of there is variation in genomics. There is large variation. So what does that mean? It means that you cannot use one person's result to generalize. A lot of things can affect the person's result. A lot of things can cause variation in the person's result. It can be their ancestral heritage. It can be the environment they grew up in. It can be the way they actually came into that disease condition. So you cannot use one replicate. You need a minimum of 3 replicates for any transcriptomics research. So you need 3 replicates per group. So for this what's it called? For this project now, we will now get 3 wild type animals and 3 5x FAD animals at any. And then the last thing is for we'll control for batch effect. So what's batch effect? So as for example, there is something that happens in genomic studies that batch effect essentially means something like that you can't control. It's more like technical error. You know, when you are doing any research, you account for technical error. Can be your machine, can be your feed that you gave your animals, can be the condition you kept them, can be the house, the animal house where you kept them, can be the cage. So those are technical things that can cause error in your results, because you need to be very, very careful that what we are measuring is actually due to genetic variation and not due to something else. Let's say, for example, now you want to study something. You want, for example, in this our research now, we have wild type animals and we have 5x FAD animals. What we can do is, okay, we want to extract the RNA. Let's say we want to do the sequencing. You have 2 sequencing machines. You have Illumina and you have PacBio. For one, what's it called? For one research, you now say, okay, for the wild type, I'll put them in the Illumina platform. And then for the 5x FAD, I'll put them in the PacBio platform. That would be a very, very wrong research. Why? Because at the end of the day, your variation might be as a result of just the difference in the platform that you use for sequencing. Might not be a real biological variation. So that's why it is very, very important that you control for batch effect. So you measuring technical variation instead of biological variation is what is called batch effect. How do you control for batch effect? One thing you can do is that you randomize every process in your research. Let's say you have 2 sequencing platforms. You can put one wild type for Illumina, one white type for you randomly pick. It's not that you put all groups in one condition and you put all the second groups in totally different conditions. It will affect your results. So it will cause batch effects. So that's how you control for batch effects. So after your experimental design, you collect your sample. So after you must have acclimated your animals for as long as you want, and you feel like, okay, you are ready to extract, you have seen the symptoms and you are sure that, okay, this thing is now present, so you want to extract the RNA. So you isolate the tissue. For this what's it called? For this case study, we are using bulk RNA sequencing. So you isolate the tissue from the mice. Then you do RNA extraction using standard protocols. So just like DNA extraction, if you are familiar with the molecular lab, there are there are kits used for extracting DNA and RNA. So you go to the you purchase the kit for the extraction. So you extract the RNA you do all the complete process and you extract the RNA. So one thing we need to understand is that RNA is very, very, very how do I put this? Is an unstable molecule, which means it degenerates rapidly. It degenerates even as. What do? You need to. That's why there is what is called RNA quality control. So you need to test the quality